In the vast, silent theater of the cosmos, the emergence of life on Earth stands as the most dramatic and inexplicable act. For billions of years, the universe was a realm of physics and chemistry, consisting only of lifeless matter governed by thermodynamics and gravity. And then, on a rocky, water-drenched planet, orbiting a mediocre star, matter began to do something extraordinary. It began to remember. The question of how life ignited from non-living matter, a process known as abiogenesis, remains the deepest mystery in science. It is a puzzle that forces us to look back 4 billion years to a Hadean Earth that was violent, toxic, and alien. For decades, scientists were deadlocked by a logical paradox, a molecular version of the chicken and egg causality loop. To understand this, we have to look at the machinery of life as we know it today. Modern life relies on a complex triumvirate. DNA stores the instructions. Proteins do the work, catalyzing reactions and building structures, and RNA acts as the messenger between them. Here lies the problem. DNA cannot replicate without protein enzymes to do the physical labor. But protein enzymes cannot be built without the instructions encoded in DNA. How, then, could life begin if both were required for the other to exist? The solution to this paradox, proposed in the late 20th century and bolstered by groundbreaking discoveries in 2024 and 2026, is the RNA World hypothesis. It suggests that long before DNA banks and protein architects, there was a versatile solitary virtuoso called ribonucleic acid, or RNA. This molecule could do it all. It could store genetic information like a hard drive, and it could catalyze chemical reactions like a machine. Today, we are going to explore the epic saga of the RNA world, tracing its history from a speculative idea to a robust scientific theory. We will delve into the molecular mechanics of ribozymes, examine the cutting-edge hunt for the holy grail of biology, which is the self-replicating RNA molecule, and discuss the revolutionary discoveries of the last few years, including the game-changing QT45 ribozyme and the systems chemistry of John Sutherland. To truly understand the revolutionary nature of the RNA world, one must first appreciate the depth of the problem it solves. By the mid-20th century, the central dogma of molecular biology was established. DNA makes RNA, and RNA makes protein. DNA is chemically stable, perfect for long-term storage. It is the genotype. Proteins, folded into intricate three-dimensional shapes, are the phenotype. They are the active agents that build cells, digest food, and copy DNA. This division of labor is efficient, but it creates an origin problem. If you strip a modern cell down to its basics, you find a factory where the blueprints and the workers are useless without each other. Spontaneous generation of a DNA molecule is statistically impossible. The spontaneous generation of a functional protein is equally absurd. Sir Fred Hoyle famously compared the likelihood of a protein forming by chance to a tornado sweeping through a junkyard and assembling a Boeing 747. For a long time, this impasse seemed insurmountable. Origin of life researchers were divided into genes first camps, believing a replicator came first, and metabolism first camps, believing a chemical cycle came first. But in the late 1960s, a few visionary thinkers began to suspect that the middle manager of the central dogma, RNA, might be the hero in disguise. Francis Crick, co-discoverer of the DNA double helix, alongside Leslie Orgel and Carl Woese, independently suggested that RNA might have been the primordial molecule. Their reasoning was based on the structure of RNA itself. Unlike DNA, which is a rigid double helix locked in a stable embrace, RNA is typically single-stranded. This flexibility allows RNA to fold back on itself, forming loops, stems, and complex 3D structures, much like a protein. If it can fold like a protein, they asked, could it act like a protein? Could it catalyze reactions? In 1986, Nobel laureate Walter Gilbert coined the term the RNA world to describe this hypothetical era. He painted a picture of a primitive Earth where RNA molecules rained down into pools, storing information in their sequence and facilitating their own replication through their folded shapes. It was an elegant solution. The chicken and the egg were the same molecule. The RNA world hypothesis would have remained a neat story if not for a seismic discovery in the early 1980s. Thomas Seck at the University of Colorado and Sidney Altman at Yale University were studying gene expression, specifically how cells process RNA. Seck was working with Tetrahymena thermophila, a single-celled pond organism. He was trying to isolate the protein enzyme responsible for cutting out a section of junk RNA from a larger strand. He purified the RNA, added it to a test tube, and removed every trace of protein. To his shock, the RNA spliced itself. The molecule was performing surgery on itself, without any protein surgeon present. Simultaneously, Altman discovered that RNase P, an enzyme that processes tRNA, was composed of both protein and RNA. When he separated them, the protein was useless. The RNA, however, could still do the work. They had discovered ribozymes, or ribonucleic acid enzymes. This shattered the biological law that all enzymes are proteins. RNA was not just a passive messenger, it was a chemical actor. For this paradigm-shifting work, Keck and Altman shared the Nobel Prize in Chemistry in 1989. But the most profound evidence for the RNA world lies deep within your own cells, inside the ribosome. The ribosome is the universal machine of life, responsible for translating RNA into proteins. It is a massive complex of RNA and proteins. For decades, biologists assumed the ribosomal RNA was just a scaffold to hold the important ribosomal proteins in place. But as X-ray crystallography improved, a startling truth emerged. In 2000, the high-resolution structure of the ribosome revealed that the active site, the place where amino acids are actually stitched together to make proteins, is made entirely of RNA. There are no proteins within 18 angstroms of the catalytic center. The ribosome is a ribozyme. This means that protein synthesis is catalyzed by RNA. At the deepest level of our biology, we are still RNA-based lifeforms using an ancient RNA machine to build our modern protein bodies. The ribosome is a molecular fossil, a relic from the RNA world that survived because it was too essential to change. If the RNA world existed, there must have been a first replicator, an RNA molecule capable of copying itself without the aid of proteins. Finding or creating such a molecule became the holy grail of experimental evolution. For 30 years, this quest was defined by frustration and incremental progress. RNA is chemically fragile. It degrades easily in water. Furthermore, copying a long strand of RNA is difficult because of the fidelity threshold. If a replicator makes too many mistakes, the errors accumulate, the information is lost, and the system collapses into an error catastrophe. In the 1990s, David Bartel and Jack Szostak used a technique called CELEX to evolve new ribozymes in the lab. They succeeded in creating the class 1 ligase, a ribozyme that could join two pieces of RNA together. This was a crucial step, but the ligase could not copy itself. It could only perform a single reaction. The real goal was an RNA-dependent RNA polymerase, a ribozyme that moves down a template strand, grabbing free nucleotides and stitching them into a new copy. By 2013, the lab of Philip Holliger had evolved the TC19Z ribozyme. This molecule could copy RNA sequences up to 95 nucleotides long. It was a masterpiece of molecular engineering, but it had a fatal flaw. It could not copy itself. It was too large and complex to be replicated by its own limited ability. The field hit a wall. To be a true self-replicator, a ribozyme needs to be precessive, accurate, general enough to copy any sequence, and small enough to be copied by its own catalytic power. However, history will likely view the mid-2020s as the golden age of origin of life research. Two separate breakthroughs, one from the Salk Institute in California and one from the MRC Laboratory of Molecular Biology in the UK, fundamentally changed the landscape. In 2024, the lab of Gerald Joyce at the Salk Institute announced a major leap forward. Joyce, a titan in the field, utilized a new form of continuous in vitro evolution. Instead of manually selecting RNA strands, his team built a system where the RNA had to survive in a test tube by constantly replicating a hammerhead ribozyme. If the polymerase made a mistake, the hammerhead wouldn't work and the line would die out. After hundreds of rounds of evolution, simulating millions of years of natural selection in a few months, they produced a polymerase ribozyme with greater than 90% accuracy. Crucially, it could evolve. When the researchers pressured the system, the ribozyme developed mutations that made it faster and more robust. Joyce called this chasing the dawn of evolution. For the first time, we had an RNA molecule that was not just a static catalyst, but a dynamic evolving entity capable of sustaining a lineage of genetic information. While Joyce's lab focused on high-fidelity polymerases, a team led by Eduardo Gianni at the MRC Laboratory of Molecular Biology took a different approach. They suspected that the previous ribozymes, which were often 200 nucleotides long, were too bloated. A primitive Earth wouldn't produce a 200-unit monster by chance. They needed something small. In early 2026, they published a paper that sent shockwaves through the scientific community. They had discovered QT45. As the name suggests, QT45 is a ribozyme only 45 nucleotides long. To put that in perspective, it is a fraction of the size of modern enzymes. Despite its diminutive size, QT45 is a powerhouse. It can catalyze the synthesis of its own complementary strand. Its small size makes it statistically plausible that such a sequence could arise spontaneously in a prebiotic pool enriched with nucleotides. And it can copy other RNA strands as well. The discovery of QT45 bridged the complexity gap. Skeptics of the RNA world had long argued that the probability of a functional ribozyme forming by chance was zero. QT45 showed that catalytic ability exists in very short, simple RNA motifs. It wasn't a Boeing 747. It was a paper airplane. Simple, functional, and capable of flight. Even with a perfect ribozyme, there is a physical problem that physics imposes on the RNA world known as strand inhibition. When an RNA strand copies a template, the result is a double-stranded RNA helix, This helix is very stable. The two strands stick together tightly. In a modern cell, protein enzymes called helicases rip these strands apart so they can be copied again. In the RNA world, there were no helicases. Once an RNA copied itself, it would be stuck to its copy, effectively dead. How did the first replicators separate? Recent work by James Atwater and others has proposed a solution involving the physical environment. The answer might be ice. In a warm little pond, the strands stick, but consider a volcanic island with freezing winters or high-altitude pools. When water freezes, it forms pure ice crystals. Impurities like RNA and salt are excluded from the ice lattice and concentrated in the tiny liquid brine channels between the crystals. This concentration helps the RNA find nucleotides and polymerize. Then, when the ice melts or when the pool heats up during the day, the water dilutes the system. Atwater's 2025 study demonstrated that using trinucleotide triphosphates, which are short 3-letter chunks of RNA, instead of single letters allowed for replication that was less prone to sticking. Furthermore, temperature cycling from hot to cold could mechanically separate the strands. The heat melts the duplex, separating the strands, and the cold concentrates them to allow copying. This suggests the origin of life may not have happened in a tropical paradise, but in a harsh, fluctuating environment, perhaps a geyser field in a snowy volcanic caldera. We have replicators, but where did the RNA building blocks come from? This was historically the weakest link in the RNA world hypothesis. A nucleotide has 3 parts, a phosphate, a sugar, and a base. For years, chemists tried to make these separately and glue them together. It never worked. The bonds were too unstable in water. Enter John Sutherland of the MRCLMB. In 2009, he revolutionized prebiotic chemistry by proposing a systems chemistry approach. Instead of making the parts separately, he threw the raw materials together. He found that hydrogen cyanide and hydrogen sulfide, 2 deadly poisons, when exposed to ultraviolet light, could undergo a cascade of reactions that produced the precursors for everything. Sutherland showed that the same reaction network produces precursors for lipids, amino acids, and nucleotides. As these reactions proceed, some byproducts crystallize and fall out of the solution, naturally purifying the remaining mixture. Recent work from his lab in 2024 has shown that this cyanosulfidic chemistry is robust. It creates chimeric intermediates, molecules that are half sugar, half base, which then fuse together perfectly to form nucleotides. This work implies that there wasn't strictly an RNA world followed by a protein world. Instead, the raw materials for RNA, peptides, and lipids likely co-emerged from the same chemical soup. The RNA world wasn't a lonely place. It was a cluttered nursery of diverse chemical species. This leads us to the concept of the RNP world, or ribonucleoprotein world. The strict definition that RNA acted alone is evolving. While RNA was likely the master architect, it probably had help from the start. Small peptides are easy to make prebiotically. Sutherland's chemistry makes them alongside RNA. Recent theories suggest a coevolution. RNA is fold-prone and sticky. Small, positively charged peptides could have bound to RNA, stabilizing its structure and helping it fold into active ribozymes. In return, RNA provided a surface for peptides to assemble. This leads to a model where the ribosome didn't just appear. It evolved from a primitive association between RNA, which handled the coding, and peptides which stabilized the RNA. The breakthrough was when an RNA discovered it could make the very peptides that helped it survive. This feedback loop is the engine that likely drove the origin of the genetic code. The study of the RNA world is not just archaeology. It is the blueprint for the future of biotechnology. Using the same CELEX technology used to find the first ribozymes, scientists now evolve aptamers, which are DNA or RNA molecules that can bind to specific targets like cancer cells. CRISPR, the gene editing revolution, is based on an ancient bacterial immune system that uses RNA guides to target DNA. The mRNA vaccines proved the power of synthetic RNA, delivering a genetic message to our cells to manufacture a viral protein, bypassing the nucleus entirely. And labs like Joyce's are not just trying to recreate the past. They are trying to build new life. By creating self-replicating RNA systems that evolve, they are on the verge of creating Life 2.0, organisms that do not use the standard DNA protein operating system. The journey from a sterile rock to a planet teeming with consciousness is a story of molecular heroism. The RNA world hypothesis provides the most compelling narrative for this transformation. The recent discoveries of QT45, the high-fidelity salt polymerase, and cyanosulfidic systems chemistry have moved the RNA world from the realm of plausible to probable. We now know that the building blocks of RNA arise naturally from simple poisons. We know that small, simple RNA sequences like QT45 can replicate themselves. And we know that RNA can evolve in a test tube to become complex, accurate, and lifelike. We are, in a very real sense, the the descendants of those first fragile ribozymes. Every time a cell in your body divides, every time a ribosome stitches an amino acid into a protein, it is an echo of a process that began in a freezing volcanic pool 4 billion years ago. The RNA world didn't end. It just built a shell of DNA and protein around itself and started exploring the universe.